Okay, so we're going to learn about DNA replication. Remember, this process happens during S phase of the cell cycle. So when the cell is ready to divide, it does DNA replication before it goes into G2 and then mitosis. So we have a lot of enzymes involved in DNA replication. This here is modeling the action of helicase, because it's actually breaking the hydrogen bonds between the DNA double helix. And then you'll notice you have two different polymerases, DNA polymerases. And that's because DNA is anti-parallel. So this strand, you can see the arrow is up here, and then this strand, you can see the arrow is up there. When you're looking at one of these nucleotides, the arrow here in this model is showing you the 3' end. So DNA polymerase can only add nucleotides to the 3' end of the DNA. So when it's like this, it actually can't start because there's no nucleotide to attach it to. So there's actually an enzyme that's going to add RNA nucleotides first. So this is going to be called the leading strand. And so to the leading strand, we're going to add just a few nucleotides of RNA. And the enzyme that does that is called primase. So it's just like adding a primer. For some reason I need a, oh, that's because I had this one, okay. All right. Oh, that's a C, so it goes with G. Okay, so now we got the primer. So primase does that. Now DNA polymerase can work because now it's got a nucleotide to attach to. And remember, it can only add to the 3' end. So the DNA polymerase is going to move towards what I call the crotch, or sometimes it's called a replication fork. So this is the leading strand. It's going to go towards the crotch. So now DNA polymerase is going to add an A here, right, and another A here, and then it's going to just move towards, so it's going to keep moving towards the helix. As this opens up more, it'll just keep going. And so it can go in one continuous strand and copy this side of the DNA. Now on the other strand, we got a whole different issue, because you can see this is the 5' end, this is the 3' end. So what we're going to have to do is we're going to have to start at the crotch and go back. So we're going to have to start by adding our RNA primer here and go this way. So let's see, we've got A, we need a U, we need a C, another C, and another U. Okay, so actually primers are more than 4 nucleotides long, but let's just say that that's our primer. So remember, primase adds the primer, and now DNA polymerase, you can see it's going to be going this way because it has to add to the 3' end. So now we have a G, we need a C. So now these are DNA. Notice that in this model, the DNA nucleotides are rounded here and the RNA nucleotides are square. So you have this RNA primer, and then we're going to add C. Remember, we're adding to the 3' end. And then we're going to be going away. Away from the crotch. Okay, so this is going to have to keep going discontinuously. And then what happens that's kind of a pain for DNA replication is as this gets more open, it's not a problem for this side because this side can keep adding nucleotides this way. But over here we're going to have to add a primer again and then add DNA in to fill it. So every single time on this is called the lagging strand, you've got to add a primer and then fill it in with DNA, and then add a primer and then fill it in with DNA. So this is going to be produced in sort of these discontinuous spurts. Then after it's all done, there's an enzyme that's going to remove the primer and replace it with DNA nucleotides. And that's called DNA polymerase 1. This enzyme is actually called DNA polymerase 3. But DNA polymerase 1 is going to remove these and replace it with DNA. Same thing over here. And then once you've done that, you've done DNA replication. The only other enzyme is an enzyme called topoisomerase, because when you open this DNA, what will happen is it will get kind of supercoiled here. And so topoisomerase breaks the backbone of the DNA, kind of like this, and lets it unwind and then glues it back together. So that's topoisomerase. Okay.